Next topic, ribosome. Ribosome in prokaryotes and eukaryotes. Ribosome structure, functional sites of ribosome, models of ribosome structure, composition of ribosome, and reconstitution experiment. What is ribosome? What are ribosome? A ribosome is a complex molecular machine found inside the living cell that produce proteins from amino acids during the process called protein synthesis or translation. The process of protein synthesis is a primary function which is performed by all living cells. Ribosomes are specialized cell organelles and found in both prokaryotic and eukaryotic cells. Every living cell requires ribosomes for the production of proteins. A ribosome is a complex of RNA and protein and is therefore known as a ribonucleoprotein. It is composed of two subunits, small and large. Discovery. Ribosomes were first discovered and observed in the mid-1950s by Romanian American cell biologist George Emil Palade using an electron microscope as dense particles or granules. Albert Claude, Christine de Dieu, and George Emil Palade were jointly awarded the Nobel Prize in Physiology or Medicine in 1974 for the discovery of ribosome. Ribosomes in prokaryotic and eukaryotic cell. Prokaryotes, ribosomes often occur freely in the cytoplasm. Usually 70S ribosomes are seen with two subunits. Eukaryotes. In eukaryotic cells, the ribosomes occur either in the cytoplasm or remain attached to the outer surface of the membrane of endoplasmic reticulum. Two types of ribosomes. Based on the sedimentation coefficient, two types of ribosomes have been recognized. 70S ribosome, comparatively smaller in size, and sedimentation coefficient is 70S. Structure of ribosome. Each ribosome is porous, hydrated, and composed of two subunits. One ribosomal subunit is larger in size and has a dome-like shape, while the other ribosomal subunit is smaller in size and occurring above the larger subunit and forming a cup-like structure. In prokaryotes, 70S ribosome consists of two subunits, 50S and 30S. 50S ribosomal subunit is larger in size and has a size of 160A to 180A. The 30S ribosomal subunit is smaller in size. The 30S ribosomal subunit is smaller in size and occur above the 50S subunit like a cap. Both the subunits remain separated by a narrow cleft. In eukaryotes, the 80S ribosome also consists of two subunits, 60S and 40S. The 60S ribosomal unit is dome-shaped with larger in size. The ribosome remain attached with the ER, nucleus, etc. The 60S remain attached with the membranes. The 40S ribosomal subunit is smaller in size and occur above the 60S subunit, forming a cap-like structure. The two ribosomal subunits remain united with each other due to the high concentration of the Mg2+ ions. When the concentration of Mg2+ ions reduces in the matrix, both ribosomal subunits get separated. Actually, in the bacterial cell, the two subunits are found to occur freely in the cytoplasm and they unite only during the process of protein synthesis. At high concentration of Mg2+, ions in the matrix of the two ribosomes become associated with each other and known as the dimer. Further, during the protein synthesis, many ribosomes are aggregated due to common messenger RNA and form the polyribosome or polysome. Polyribosome or polysome, 70S ribosome and 80S ribosome. Prokaryotic ribosomes and eukaryotic ribosomes. Prokaryotic 50S subunit and 30S subunit, 70S. 60S subunit and 40S subunit, 80S eukaryotic ribosome. Functional sites of ribosomes. There are three binding sites on each ribosome. They are H site, P site and E site. A, P, E. The A site, the amino acyl site or A site binds the incoming amino acyl tRNA. P site, the peptidyl site, binds the tRNA carrying the growing polypeptide chain. E site, the exit site, binds the departing uncharged RNA. The smaller subunit has a binding site for mRNA. The codons on the mRNA determine the type of tRNA binding with A and P sites. Look at this picture. 50S subunit and 30S subunit. P site, E site, A site, amino acyl site, peptidyl site and E site, exit site, mRNA binding site. Then mRNA, along with that mRNA binding site, growing polypeptide chain, A site, P site, E site, 50S and 30S. Models of ribosome structure. Two models have been proposed to explain the structure and mode of association of subunits of bacterial ribosomes. They are quasi-symmetrical model proposed by Stoffler and Witzmann, then the asymmetrical model proposed by Lake. Quasi-symmetrical model. According to this model, the larger subunit is almost bilaterally symmetrical and the smaller one irregular. The larger subunit has a spherical base and 3 protuberants. It is comparable to an armchair that has a circular seat. Its spherical base forms a seat for the simpler subunit. The prominent central protuberance forms the corresponding back and the lateral ones form the armrest. The smaller subunit is slightly bent and bipartite rod comparable to a telephone receiver. It has a cleft which divides it into the smaller head and large body or base. Body has elongated extensions on both the sides. The long axis of the smaller subunit is placed at right angles to the central protuberance on the larger ones, and its cleft directly faces the vaulted seat of the larger subunit. As a result of this association, a gap or groove is left in between the two subunits for the orientation of mRNA. Central protuberance, body, 30S subunit, larger. Lateral crest or arms, lobe region, vaulted seat, 70S subunit, base, tunnel, cleft, head and dent. Lake's asymmetrical model. According to this model, both the subunits are asymmetrical. The larger one has a spherical body or base and 3 major protuberants, one central and the other lateral. One of the lateral protuberants is long and stalk-like and the other one is usually only a ridge. The smaller subunit has an indentation which divides it into upper body and upper head and lower body or base. Extending from the base is a small platform. In between the platform and the head is a cleft. It is a site for codon-anticodon interaction and also for the binding of the initiation factor of protein synthesis. The smaller subunit is positioned over the larger one and its head is aligned to the central projection of the larger one. A groove is left in between them for the orientation of mRNA. Head, base, cleft, platform, valley, ridge, central protuberance, stalk. Base, smaller subunit plus larger subunit, ribosome. Head, platform, base, small subunit plus large subunit, ribosome. Composition of ribosomes. Ribosomes are made up of ribosomal proteins and ribosomal RNA. Ribosomes are usually made up of 3 or 4 ribosomal RNA molecules, anywhere from about 40 to 80 different ribosomal proteins. Each ribosome is composed of 2 subunits, a larger one and a smaller one, each of which has a characteristic shape. Ribosomal RNA. The 70S ribosome contain 3 types of ribosomal RNA: 23S rRNA, 16S rRNA and 5S rRNA. The 23S and 5S rRNA occur in the larger 50S ribosomal subunit, while the 16S rRNA occurs in the smaller 30S ribosomal subunit. Assuming an average molecular weight for one nucleotide to be 330 Dalton, one can calculate the total number of each type of rRNA. Thus, the 33S rRNA consists of 330 nucleotides, 16S rRNA contains 1650 nucleotides. The 80S ribosomes contains 4 types of rRNAs, which are 28S rRNA, 18S rRNA, 5S rRNA and 5.8S rRNA. The 28S rRNA occur in large 60S ribosomal subunit. About 60% of the RNA is helical and contain paired bases. These double-stranded regions are due to hairpin loops between the complementary regions of the linear molecule. The 28S rRNA has the molecular weight of 1.6 to 1.1 into 10 raised to 6 Dalton, and its molecules are double-stranded and having nitrogen bases in it. Ribosomal proteins. Each 70S ribosome of E. coli is composed of about 55 ribosomal protein. Out of this 55 protein, about 21 different molecules have been isolated from the 30S ribosomal subunit and some 32 to 34 protein from the 50S ribosomal subunit. The primary structure of several of these proteins has been elucidated. Most of the recent knowledge about the structure of the ribosomal protein has been achieved by the dissociation of ribosomal subunit into their components rRNA and protein molecules. Metallic ions. The most important low molecular weight components of ribosomes are the divalent metallic ions such as Mg2+, Ca2+, and Mn2+. Then reconstitution experiment. When ribosomes are centrifuged in a gradient of 5 mCsCl, about 30 to 40% of the protein is lost. Simultaneously, some other proteins called split proteins are released from the ribosome. When the split proteins are added to their corresponding core particles, the ribosomes are reconstituted. Some of the split proteins are apparently specific for each ribosomal subunit. The split proteins have been further fractioned and divided into acidic and basic protein. Nomura et al. Fractioned at least 6 different groups of proteins in the ribosome. The reconstitution of the functional 30S ribosome subunit has been achieved by the addition of 30S rRNA to a mixture of proteins from 30S subunit. Following a similar procedure, the 50S subunit has also been reconstituted. Along with the subunit, the whole ribosome is also assembled. In vitro reconstitution is a powerful tool for investigating ribosome functions and biogenesis as well as discovering new ribosomal features. Next topic, chaperones and functions. What are chaperones? Chaperones are Chaperones are a functionally related group of proteins that assist the covalent bonding or unfolding and the assembly or disassembly of other macromolecular structures. They are found in all kinds of cells and cellular compartments and have a wide range of binding specificities and functional roles. Bind non-native proteins preventing unspecific aggregation. Landmark feature of molecular chaperones is the involvement of energy-dependent reaction in the folding process. The misfolded or unfolded polypeptide chain to chaperones bind are said to be non-native as they are not folded into their functional conformations. Non-stress native protein, unfolded protein, unfolding stress aggregate, chaperones and substrate complex. Inactive ATP independent chaperones, active chaperone substrate complex, non-stress. Refolded protein, ATP dependent chaperones, inactivation and release. So the molecular chaperones are a diverse group of families of proteins that are required for their direct folding, transport, or degradation of other proteins in vivo. They are formed with amino acid monomers. They do not interact with the native proteins, or they do the part of the final folded structures. Chaperones are present when the macromolecules perform their normal biological functions, complete the process of folding and assembly. The chaperones are concerned primarily with protein folding. Some chaperones are non-specific and interact with a wide variety of polypeptide chains, but others are restricted to specific targets. In humans, mostly in endoplasmic reticulum, their location. General chaperones are given. Lectin chaperones, calnexin and calreticulin. Non-classical molecular chaperones, HSP45 and ERP29. Folding chaperones, protein disulfide isomerase, peptidyl prolysis trans isomerase. Mechanism. DNAG binds to the unfolded or partially folded protein and then to DNAK. DNAJ stimulates ATP hydrolysis by DNAK. DNAK ADP binds tightly to the unfolded protein. In bacteria, the nucleotide exchange factor GRPE stimulates release of ADP. ATP binds to DNAK and protein dissociates. Properties of chaperones. Molecular chaperones interact with unfolded or partially folded protein subunits. Nascent chains emerging from the ribosome or extended chains being translocated across subcellular membranes. They stabilize non-native conformation and facilitate correct folding of protein subunits. They do not interact with native proteins, nor do they form part of the final folded structures. Some chaperones are non-specific and interact with a wide variety of polypeptide genes, but others are restricted to specific targets. They often couple ATP binding or hydrolysis to the folding process. Essential for viability, their expression is often increased by cellular stress. Functions of chaperones. They act as a container for the folding of other protein subunits, as they are called heat-shocked proteins. They also prevent the degradation of proteins in spinal stress conditions. Some chaperone systems work as foldases. They support the folding of proteins in an ATP-dependent manner, for example in the GRPE or the DNA-K, DNA-J, GRPE system. Other chaperones work as holdases. They bind folding intermediates to prevent their aggregation, for example DNA-J or HSP33. Chaperones work in coordination by forming assemblies, such as assembly chaperones, especially in the nucleus, are concerned with the assembly of folded subunits into oligomeric structures. The first protein is to be called a chaperone assists the assembly of nucleosomes from folded histones and DNA. They recognize and correct mistakes in folding by binding to the non-polar surfaces, promote correct folding of their substrate proteins by unfolding incorrect polypeptide chain conformations, providing an environment in which correct protein folding can occur. As heat shock proteins. Heat shock proteins are a family of proteins that are produced by cells in response to exposure to stressful conditions. Several heat shock proteins function as intracellular chaperones for other proteins in bacteria like E. coli. Chaperones are highly expressed under high stress, for example, high temperature. For this reason, the term heat shock protein has historically been used to name these chaperones. The prefix HSP designates that the protein is a heat shock protein. The reason for this behavior is that protein folding is severely affected by heat, and therefore some chaperones act to prevent or correct damage caused by misfolding. Then macromolecular crowding. The crowded environment of the cytosol can accelerate the folding process, as a compact folded protein can occupy less volume than an unfolded protein chain. Crowding can reduce the yield of correctly folded protein by increasing protein aggregation. Crowding may also increase the effectiveness of the chaperone protein such as GRPE, which could counter-react to the reduction in folding efficiency. Cell homeostasis, two opposite functions, protein folding and degradation. The two processes are carried out through the transient formation of complexes between different chaperones and co-chaperones. Transport across membrane of the mitochondria. Endoplasmic reticulum bacterial translocation specific chaperone maintains newly synthesized precursor polypeptide chains in a translocation competent system and guides them to translocon. New functions of chaperones. New functions of chaperones continue to be discovered, such as assistance in protein degradation, bacterial adhesion activity in responding to diseases linked to protein aggregation and cancer maintenance. Main role, they prevent inappropriate association or aggregation of exposed hydrophobic surfaces, direct their substrates into reproductive finding, transport, or degradation pathways. The type 1 intermolecular chaperones, first discovered based on the studies on subtilisin, an alkaline serine protease from Bacillus subtilis, mediate the folding of proteins into their respective tertiary structures and are mostly produced as the N-terminal sequence extension. Then type 2 intramolecular protein mediate the formation of the quaternary or functional structure of proteins usually located at the C-terminus of the protein type 2 intramolecular chaperones. Mutations in the intramolecular chaperones can cause misfolding of the functional domain, results in a distortion of their function, leading to human diseases. Alzheimer's disease, Parkinson's disease, mad cow disease, and transthyretin amyloidosis. Small heat shock proteins, HSP25 holders. HSP25 is the second largest of the 16 identifiable small heat shock proteins in the nematode, protect against cellular stress, prevent aggregation in the lens. Attract. HSP 90 ATPase holder. HSP 90 is a specialized chaperone that assists in the maturation of client proteins. These proteins include over 100 transcription factors and kinases such as steroid receptors. HSP 70 chaperones. Their size is approximately 70,000 Daltons. Best characterized small chaperones often work in concert with one or more smaller co-chaperone proteins which serve to modulate the activity of the chaperone. HSP 60 chaperones. HSP 60, also called chaperonins, are barrel-shaped structures composed of 14 to 16 subunits of proteins that are approximately 60,000 Dalton in size and best characterized large chaperone complex. Each subunit has a patch of non-polar amino acid groups lining the inner surface of the barrel. This patch recognizes the composed non-polar amino acid of misfolded protein. The binding and hydrolysis of ATP triggers conformational changes within the barrel. Hollow cylinder polypeptide correctly folded protein. An unfolded polypeptide enters the cylinder from one end. The cap attaches, causing the cylinder to change shape in such a way that it creates a hydrophilic environment for the folding of the polypeptide. The cap comes off and the properly folded protein is released. Then GROEI chaperone. GROEI chaperone consists of two rings, cis or proximal upper ring and trans or distal lower ring. GROES co-chaperone binds to both GROES rings. Each of the GROEL rings consists of 7 identical units shown in the tower. The units are arranged in a circular manner to form a cavity. GROES upon binding to GROEL serve as lid which encapsulates the volume inside the cavity. The top view of GROEL ES-GROEL bacterial chaperone complex model. Chaperones and human disease. It is clear that molecular chaperones assist with the folding of newly synthesized proteins and correct protein misfolding. Recent studies now suggest that defects in molecular chaperone substance interactions may also play a substantial role in human disease. Next, ribosome structure. Active centers in ribosome. Ribosome and protein synthesis. Biogenesis of ribosome in eukaryotes. So, introduction. The ribosome is a complex molecule made of ribosomal RNA molecule and proteins that form a factory of protein synthesis in cells. In 1955, George E. Palland discovered the ribosome is a complex molecule made of ribosomal RNA proteins and form a factory of molecular synthesis in cells. In 1955, George E. Palland discovered ribosomes and described them as small particles in the cytoplasm that preferentially associate with the endoplasmic reticulum membrane. Ribosomes have two main functions, decoding the message and the formation of peptide bonds. These two activities reside in two large ribonuclear protein particles, RNPs, of unequal size, the ribosomal subunits. Each subunit is made of one or more ribosomal RNAs or rRNAs, as many ribosomal proteins are proteins. Ribosomes are organelles located inside the animal, human cell and plant cells. They are situated in the cytosol, some bound and free-floating to the membranes of the coarse endoplasmic reticulum. They are utilized in decoding DNA, deoxyribonucleic acid, two proteins, and no rRNA is forever bound to the RER. They release or bind as directed by the kind of protein they proceed to combine. In an animal or human cell, there could be up to 10 million ribosomes, and numerous ribosomes can be connected to the equivalent mRNA strand. The structure is known as a polysome. Then ribosome, nucleus, ribosome, endoplasmic reticulum, growing peptide chain is there, mRNA, ribosome, small subunit, amino acid, ribosome, large subunit, tRNA, large subunit, ribosome, mRNA, polypeptide chain, eukaryotic polysome. Ribosome structure. Ribosomes are made of proteins and ribonucleic acid abbreviated as RNA in almost equal amounts. It comprises of two sections known as subunits, large and small subunit. The thinner subunit is in place the mRNA binds and it decodes, whereas the bigger subunit is the place the amino acids are included. Both subunits comprise of both ribonucleic acid and protein components and are linked to each other by interactions between the proteins in one subunit and RNAs in the other subunit. The ribonucleic acid is obtained from the nucleolus at the point where ribosomes are arranged in a cell. Eukaryotic ribosome and prokaryotic ribosome 50s, 30s, 70s, eukaryotic ribosome 60s, 40s, 80s. Structure of ribosome mRNA, large subunit, tRNA docking sites, small subunit, mRNA binding site, modeled 3D and specified one. Ribosome active centers. There are mainly 4 binding sites for the binding of mRNA and tRNA. In addition to this, there is extra 2 site also can be seen, which is the peptidyl transferase center and translation factor binding site on the large subunit. tRNA, protein chain, amino acid, ribosome, mRNA and movement of ribosome. M site. This is the mRNA binding site on the small subunit. The ribosome has intrinsic affinity for template polynucleotide. It is located on the small ribosomal subunit in the region of groove dividing the head, the body and side bulge of it. These proteins such as S1, S2, S5 can be seen in mRNA binding sites. Protein S1 occupying the interline position on the lateral surface of the small subunit in the region of the neck. It has an ability in forming complex with polynucleotide. Binding of this isolated protein S1 with mRNA results in the loosening or unfolding of mRNA secondary structure. Next, protein S5 is located in the 30S subunit head near the neck and the S3 opposite to that of S5. Then peptidyl transferase center PTC. Catalysis of peptide bond formation. There is a peptidyl transferase center in the large subunit of ribosome, which contains 2 substrate binding sites, acceptor binding site and donor binding site, known as D and A sites of PTC of several peptidyl transferase activity. It is the main and seemingly the only catalytic function of the ribosome itself. It is responsible for the formation of peptide bonds during polypeptide elongation. In the translating ribosome, transpeptidation proceeds between the peptidyl tRNA and amino acid tRNA. In this reaction, the peptidyl tRNA serves as the donor substrate and the amino acid tRNA and add the acceptor substrate. During transpeptidation, the PTC binds the 3' terminal adenosine with the amino acid residue of the A site bound tRNA at the A site and 3' terminal CCA sequence with amino acid residue and its peptide group of the P site bound tRNA at the D site. PTC is located at the large subunit on its interface side under the head and more exactly in the region of the groove separating the head from the rest of the body. There have been many attempts in isolating the enzyme from large subunit, which is responsible for catalyzing interpeptidation. During transpeptidation, the PTC binds the 3' terminal adenosine with the amino acid residue of the A site bound tRNA at the site and the 3' terminal CCA sequence with amino acid residue and its peptide group of the P site bound tRNA at the D site. Factor binding site. GTP dependent binding of translation factors occurs here. It is located on the large subunit.